Hello everyone. Today we want to talk about protein function, and to illustrate these concepts we will talk about hemoglobin and myoglobin. We are using a case called The Mermaid and the Globins. It was published by the National Center for Case Study Teaching and Science, and we answered questions 1 and 2 were reported out on Perusall. So I will direct you to Perusall if you'd like to see your classmates' comments. So let's go on. Myoglobin and hemoglobin have different physiological functions. Myoglobin stores oxygen in muscle cells by facilitating diffusion of oxygen throughout the cell, whereas hemoglobin is an oxygen transporter in blood. So they have different physiological functions, and they're found in different tissues. So they must have something in common because they can both bind oxygen, but there must be some differences too. So that means we have to take our usual structure-function approach to figure out why. So let's start with myoglobin first because it's the simpler of the two proteins. And let's go through the primary and secondary and tertiary structure. So the primary structure, myoglobin, is a single chain 153 amino acid protein. There are no disulfide bonds, as is typical in proteins that are found inside the cell. In terms of secondary structure, myoglobin is 75% alpha helical. There are no beta sheets. And the people who study myoglobin label these helices A through H. We can sort of trace through A, B, C, D, E, F, G, H throughout this protein. You also might have noticed in the center in red is a prosthetic group. So I have some close-up pictures of that I'll show you in the next slides. So the tertiary structure, the three-dimensional structure, is shown in the figure. So let's zero in on that prosthetic group. So the heme group consists of two components, an iron 2+ ion and protoporphyrin 9. So the protoporphyrin 9 ring is shown in black. It's a planar aromatic ring. And in the center, in red, is an iron ion, and the iron has to be in the +2 oxidation state in order to bind oxygen. The iron can form 6 coordinate covalent bonds. It can form 4 bonds to nitrogen, so we have 1, 2, 3, 4 bonds here shown in red. 1 bond will be to the nitrogen of histidine F8, so that's the 93rd residue in the entire chain. F8 means it's the 8th residue in the F helix. So that's called the proximal histidine, and it will form that 5th coordinate covalent bond. And this dotted line right here is the 6th bond, which can form with oxygen. So that 6th coordinate covalent position will be occupied by oxygen if myoglobin is oxygenated, and it will be empty if it is not. So here's a close-up picture right here. So the protoporphyrin 9 ring is shown with the carbons in gray. In the center right here is a purple iron ion, and here we can show the 6 coordinate covalent bonds. So there's 1, 2, 3, 4 to the nitrogen shown in blue. Here's the 5th one, and that's to the nitrogen in the proximal histidine shown down here in the lower right-hand corner. Proximal means close by. If we go up here, the iron, again, the purple iron, the 6th coordinate covalent position is occupied by oxygen, which binds it a little bit at an angle, and it's shown here in red. And the oxygen forms a hydrogen bond, so that's shown in this dashed yellow line right here, and that's called the distal histidine. So proximal histidine and distal histidine, proximal means close by, distal means further away. Distal histidine is histidine E7. So again, that's the 64th amino acid residue of the 153 in myoglobin, and it's the 7th residue in the E helix. So remember, oxygen binds reversibly as long as the iron is in the F2, the 2+ oxidation state. The protein chain prevents the, or at least discourages the oxidation of Fe2+ to Fe3+, because Fe3+ would not bind to oxygen. So here's another close-up picture right here. So in the center in red, we show the protoporphyrin 9 ring. Here, the artist has drawn the iron in the center in this brown color, and the oxygen is shown in green. And here we see the proximal histidine, histidine number 93, that will form a bond, which is actually not shown in this figure. And the distal histidine will form a hydrogen bond with the oxygen. The other things shown in this figure on either side, there's a valine 68 and a phenylalanine 43. These are hydrophobic amino acids, so this shows that there's a hydrophobic cleft in which the protoporphyrin 9 ring fits, which makes sense because this is a hydrophobic planar aromatic compound. So if you look at the tertiary structure, so this picture over here on the left shows, here's the protoporphyrin 9 ring right here, and it shows that the artist here shows the iron in purple. And this shows how the heme fits into this narrow hydrophobic cleft in the myoglobin. Now imagine we take this figure on the left and we do a cross section. Maybe we take a razor blade and we slice it in half. And we can see, if we look over here on the right, the hydrophobic amino acids are shown in yellow, and the hydrophilic amino acids are shown in blue. So you see what we'd expect, that the hydrophobic amino acids are largely found in the center of the folded protein, and the hydrophilic amino acids are found on the outside. So here's another view. So over on the left-hand side, we can see this is a space-filling view. This doesn't allow us to see the alpha helices very well, but at least it does show the idea that this protein doesn't have a lot of empty space. If we look at this picture over here on the right with the helices, we can see the helices very well, but it gives us maybe the erroneous impression that there's a lot of empty space. There isn't. Again, if I'm looking over here on the left, here in purple, you can see that's the cleft where the protoporphyrin 9 ring fits in. So this is a space-filling model that shows the electron clouds, whereas this ribbon diagram just shows the conformation of the protein chain itself and doesn't show any of the amino acid side chains. So myoglobin's physiological function is as an oxygen storage protein in muscle cells, but its main physiological role is to facilitate oxygen diffusion in the muscle. It sort of acts like a molecular bucket brigade to deliver oxygen to the respiring muscle. So a respiring muscle will use glucose, and it requires oxygen. So in the presence of oxygen, glucose can be completely oxidized to CO2 and water and provide quite a lot of ATP for the respiring muscle. So next, I'd like to derive the oxygen binding curve of myoglobin from the equilibrium constant. So here we write the equation. This is myoglobin with oxygen bound, and then we write the dissociation. So the oxygen here is shown dissociated. So we're assuming here that we have a single binding site. So if you were Max Perutz and you were the first person to determine the oxygen binding, the three-dimensional structure of myoglobin, you wouldn't necessarily know that. But you can derive this equation assuming we have a single oxygen binding site. From there, we can write the equilibrium constant expression. So that's just the molar concentration of products divided by the molar concentration of reactants. So here, I'm just going to do some rearranging. I cross multiply, and then I solve for myoglobin. So I'm going to come back to this in a minute. So what I want to do first, though, is I want to define the saturation. So that's designated as a capital Y. So the saturation Y is defined as the fractional occupancy of oxygen binding sites. So that means that Y is equal to the binding sites occupied over total sites. So for myoglobin, that means that the total sites are myoglobin that has oxygen bound plus myoglobin that doesn't. That's the total sites. And then MbO2 right here are the binding sites occupied. Okay, so here's my definition of Y, and here's my rearranged equilibrium constant expression from the slide before that. So I'm going to take this entire expression right here and substitute that in for myoglobin, and now I have something that looks more complicated, but I have expressed everything in terms of MbO2. Okay, so to continue, we we'll take this expression and we will divide through by MbO2 to simplify this fraction, and then we'll take this as a complex fraction, so we'll simplify that. and we end up with Y equals the molar concentration of O2 divided by the molar concentration of O2 plus K. So we don't typically measure the concentration of gases as moles per liter. We could, but usually we use partial pressure. So PO2 is partial pressure, and we usually use torr or maybe millimeters of mercury. So I'm going to substitute molar concentration for PO2. I'm going to make one more substitution. We're going to define K as P50, and P50 will be defined as that partial pressure at which myoglobin is half saturated with oxygen. So now we have our final equation. Y equals PO2 divided by PO2 plus P50. You might have noticed that's the equation of a hyperbola. So the equation of a hyperbola is y equals ax over b plus x. So that means that a is defined as the asymptote of this hyperbolic curve, y at infinite x. B is a point on the y-axis corresponding to a divided by 2. So if we plot the oxygen partial pressure on the x-axis and saturation y on the y-axis, we get our hyperbolic curve. So this is the curve that you get if you go into the lab with your purified myoglobin in an oxygen canister and you measure the oxygen binding at various oxygen partial pressures. But you see it's also the curve that we get when we derive the equation, assuming we have a single binding site. So when the experimental and the theoretical match, that means the experimental results confirm the theoretical derivation. Our understanding of how oxygen binds to myoglobin is correct. There's only one oxygen binding site. So what that means then is we can use this equation and we can show that when y is equal to 0, we have no oxygen bound to myoglobin. Here's our definition of P50. So that when the oxygen partial pressure is equal to P50, half of the sites are occupied. When we get to quite high oxygen partial pressures that are much, much greater than the P50, all or nearly all of the sites are occupied. So there's our curve. So there's our equation. So we can see that at low oxygen partial pressures, the saturation Y is small, and the curve here is almost linear. Not a lot of oxygen is bound to myoglobin. But when we get to much higher oxygen partial pressures, we get here, there's our asymptote is 1. So virtually all myoglobin has oxygen bound. So what we did in class is we obtained page 3 from our group manager. Actually, I put it on Sakai in your Dropbox. So we discussed the answer to question 3 in our teams. And that was, what is the P50 for myoglobin? Well, all we need to do is look at the definition. When y is 0.5, we draw a line over and drop a line down. And we find out that the P50 for myoglobin is equal to 2.8 torr. So that means that when the oxygen partial pressure is 2.8 torr, half of the myoglobins have oxygen bound. So that's a fairly low value. I only have to get the oxygen partial pressure to 2.8 torr for myoglobin to have half of its binding sites occupied. So this low P50 indicates a high affinity for oxygen. And in fact, that's what we're going to use the P50 value to mean. It's an indication of oxygen binding affinity. So again, the P50 is that oxygen partial pressure at which half of the oxygen binding sites are occupied. And if that value is low, we don't need to get the oxygen partial pressure very high for half of the binding sites to be occupied. So we did question 4 of the case study, and again, you can go back and look in your Dropbox folders if you'd like to look at that again. We can use our equation, and we can do these calculations, and we can see that y, when the oxygen partial pressure is 30 torr, y equals 0.91, and when the oxygen partial pressure is 100 torr, y equals 0.97. So there's not really a whole lot of difference between those two. The reason those values were chosen is because 30 torr is about the oxygen partial pressure in tissues, whereas 100 torr is the oxygen partial pressure in the lungs. Now, we don't have myoglobin in the lungs, but that's the reason why these were chosen. So that means that whether the oxygen partial pressure is low at 30 torr or high at 100 torr, most of the oxygen binding sites of myoglobin are occupied. All right, let's go on to hemoglobin. Hemoglobin consists of 4 chains, so it's more complicated than myoglobin, which only consists of 1. We call them alpha chains and beta chains, so that's just the nomenclature that biochemists use. It has nothing to do with alpha and beta pleated sheets, so that's something different. So we just say 2 of them are alpha chains. They're identical. They have 141 amino acids, and 2 are beta chains, which are 146 amino acids. So a little bit smaller than myoglobin, but about the same size. And if we look at the secondary and the tertiary structure of both the alpha and the beta chains and myoglobin, we'll see that they're surprisingly similar, especially when you look at the primary sequences and they're not that similar. So that might sort of fly in the face of what we've said earlier, that the primary structure determines tertiary structure. But this just goes to show that there are different primary sequences that it can assume the same tertiary structure. So we have 4 chains, and each chain has a heme, so hemoglobin can bind 4 oxygens. So here's our picture right here. So we have our alpha and our beta chains, and in red, in the center of these chains is our heme group. So you can kind of barely see this one right here. So there's 1 here, 1 here, 1 here, and 1 here, and they're all shown in red. So hemoglobin is a tetramer. It has quaternary structure, whereas myoglobin didn't because it only had 1 chain. So we use alpha 2, beta 2. That's the stoichiometry. So 2 alpha chains that are identical and 2 beta chains that are identical to each other, but not the same as the alpha chain. So what's interesting about having more than 1 chain is that the alpha and the beta chains interact with each other in such a way that the oxygen binding properties of hemoglobin are different from those of myoglobin. We'll see some of that today, but mostly we'll investigate that next week. So if we do the same thing where we derive the oxygen binding curve for hemoglobin, here we're going to assume multiple binding sites. So we'll just say HbO2 to the N, so we have N number of oxygens bound, and then we'll show the dissociation. And then we'll do the same thing that we did before. We'll write the equilibrium constant expression. So you might remember from general chemistry, if you have a coefficient, you'll raise it to that power. So I'm going to write the definition of Y now for hemoglobin. So remember, the saturation Y was oxygen binding sites divided by total sites. So HbO2 to the N, that's oxygen bound, and just hemoglobin is just oxygen's not bound, but that's the total sites in the denominator, and then hemoglobin with oxygen bound is in the numerator. So I've decided not to go through the entire derivation for you, but you can do it for yourself. And if you do that, you get this equation right here. So we have PO2 to the nth power divided by PO2 to the nth power plus P50 to the nth power. It's really, though, the same equation as it is for myoglobin. That's the equation we got for myoglobin. So if we just say that both of the equations are the same, we can just say, well, this equation right up here is the same, except for myoglobin, N is equal to 1, whereas for hemoglobin, N is greater than 1. So if we use this equation, then if N is greater than 1, it's not a hyperbola anymore. That's not the equation of a hyperbola. In fact, you get a sigmoidal or an S-shaped curve instead. So that's what you see. If N is equal to 1, you get a hyperbolic curve. If N is greater than 1, you get a sigmoidal curve. And it looks like this. So we can do the same thing where we can figure out what hemoglobin's P50 value is. So we do the same thing. So that was question 5 that we answered in class. So if you do the same thing where you drop a line over at 0.5 and drop a line down, you get a P50 value of 26 torr. Now that's quite a bit higher than it was for myoglobin. So this high value indicates a low affinity for oxygen. This means that we need to get the oxygen partial pressure all the way up to 26 torr before we have half of our binding sites occupied. The sigmoidal curve indicates that there are multiple oxygen binding sites, so we've already seen that, but it also means that the binding of oxygen to hemoglobin is cooperative. What that means that binding of the first oxygen ligand makes it easier for subsequent ligands to bind. So there are a number of analogies for this. So one is, this is from Stryer. So this is sort of Stryer's little joke because he has stamps with William Harvey on it. So William Harvey was the discoverer of the circulatory system. So if you had these sort of hypothetical stamps, if you wanted to release the first stamp, you would have to tear 2 perforations. Releasing the second stamp only means you have to tear 1. And then for the third one, you also have to tear 1, and then to release the fourth one, you don't have to tear anything because it's already it's already free. So the seniors might remember before the renovation, this is the biology department office, and for what reason, I don't know why, there is a framed picture of William Harvey, and many, many generations of Providence College students have walked by that picture and not realized who that was. I don't know where that picture is now that we've done the renovation. So we need to be able to discover what that n value is. So we'll use the Hill equation to figure that out. So again, if n is equal to 1, there's no cooperativity. The binding curve is a hyperbola, like we saw for myoglobin. If n is greater than 1, that means that there is cooperativity. And what that means is binding of one ligand makes it easier and easier for subsequent ligands to bind. And then you get a sigmoidal curve that indicates that. So if I go back to my equation that showed the sigmoidal curve, and if I do some mathematical transformations that I'm not going to show here to transform it into a straight line, right here, y equals mx plus b, what I can do is I can plot the log of y divided by 1 minus y on the y-axis, and then I can plot log of PO2 on the x-axis, and then my slope is n. So that's how I figure out what n is. And if I do that, there's a curve right here. So there are lower and upper asymptotes. So here, the slope for hemoglobin is about 3. If you do this for myoglobin, your slope is equal to 1. So for myoglobin, n is equal to 1. You get a hyperbola. There's no cooperativity. There's no one to cooperate with. You only have one chain. But for hemoglobin, n is about equal to 3, depending on some physiological circumstances, and that means you have cooperativity. So the next thing we did in class is we answered question 6 in our teams, and that meant going back to this equation and calculating y at 30 torr and 100 torr for hemoglobin. And here we have to raise this to the 3rd power. When we do this equation, we get y is equal to 0.6 at 30 torr and y equals 0.98 at 100 torr. So now we get a much larger difference in y between the low and the high partial pressures of oxygen. So this represents the amount of oxygen that's released from hemoglobin. So hemoglobin is in the blood, and say at the longest partial pressure of that's about 100 torr, then 98% of the hemoglobin has oxygen bound. When it circulates to the tissues where the partial pressure is much lower, at 30 torr, now only 60% of the hemoglobin has oxygen bound. That means it's delivered, almost 40% of its bound oxygen to the tissues. And that's what we want. We want hemoglobin to be delivering oxygen to the tissues. So what that means then is oxygen released by hemoglobin is bound by myoglobin. If we look at this red arrow right here on the right side, at 100 torr in the lungs, hemoglobin binds oxygen, and it's 98% oxygen bound. When it gets to the tissues, then it releases its oxygen, and it delivers the oxygen to myoglobin, because you might remember that myoglobin had a Y value of 0.91 at the lower oxygen partial pressure. So we can transfer our oxygen from the hemoglobin to the myoglobin, and we deliver oxygen to the cells that need it. So this is the oxygen binding curve that's shown in our case study. So if you look at the sigmoidal curve right here that's in blue and compare it to another protein that has the same P50, so if you draw a line over and draw a line down, it has the same P50. But if you look at our our blue line right here, we can see that at 100 torr, hemoglobin is nearly saturated. If you get down to the oxygen partial pressure in the tissues, which could vary anywhere between, say, 20 to 400 torr. So here, if we look at, say, 20, we deliver a whole lot more oxygen. So there'd be the difference between the 100% here and, say, at 20 torr right here, like that much oxygen is being delivered. If we looked at suppose we were in some other alternate universe where hemoglobin wasn't cooperative, you can see that this hemoglobin isn't nearly as saturated in the lungs, and it delivers far less. Again, if we look over here at 20, it delivers far less of the oxygen that it did have. So luckily for us, hemoglobin binds hemoglobin and releases it cooperatively. So page 5 of our case study is posted on perusal, so read that document and make some comments regarding questions 10 and 11. We'll discuss that in class, and then we will continue with our case study. So I hope this is helpful, and I will see you in class.